From the JAMA Network, this is JAMA Ophthalmology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Ophthalmology.
Hello, and welcome to this author interview from the JAMA Network. I'm Dr. Neil Bressler, Editor-in-Chief of JAMA Ophthalmology, and today we're going to look at the risk of new retinal vascular occlusions following mRNA COVID-19 vaccinations with enlarged aggregated electronic health record data. And joining me is one of the senior authors of this investigation, Dr. Rishi Singh. Dr. Singh is a staff physician and president of the Cleveland Clinic, Florida. Rishi, thank you for joining us.
Thank you for having me, Neil.
Maybe we just could start with why you and your colleagues decided to investigate this in the first place.
Yeah, Neil, when the first time when COVID vaccinations came about and COVID-19 came about, we had these random case reports that were coming out of many different sources, both for cerebrovascular events following COVID vaccinations. We had the discussion around retinal vein occlusions, retinal artery occlusions. There was just a plethora of studies. And we know that there was some obvious thought around the hyperviscosity state after an mRNA vaccination that might be considered theoretical, but yet could be a risk for developing these. And so we wanted to investigate this in a very larger data set to understand better whether it truly was an association or not. And we looked to see what other data sets might be available so we can ask and answer this sort of question.
It seems very important. I know that when people would report that somebody had a mRNA vaccination, and then a week later they had a retinal vein occlusion, we would say, well, wait a minute, people used to come in with retinal vein occlusions all the time before these vaccinations, and now there's hundreds of millions, maybe over 1 billion of the vaccinations that were given. And so these individual reports just wouldn't tell us it. And I think that's why, at least from JAMA Ophthalmology's perspective, we said, we're not interested in those individual case reports by and large. It really is going to have to be some large epidemiologic study. So how large was your study?
So in this group of individuals, which we got from the Trinetics platform, this is a federated aggregated electronic health record research network for de-identified data across multiple EHR systems. There's 103 million patients. And we're able to look at about 3 million of those patients who received an mRNA vaccine during the time of COVID and had potentially the diagnosis of a new RVO within 21 days of vaccination. And so we looked at those patients and compared those patients in particular to those typical vaccinations that we're all used to seeing in our populations, influenza, tetanus, diphtheria, and pertussis. Those vaccines are not mRNA vaccines, they're protein-based vaccines. And some people feel that there might be an increased risk just by the mRNA alone. And that's why we wanted to look at that and see if there was a difference in the two vaccination types potentially, and see if that might have led to an increased rate of RVO in those populations.
So you're looking for who has this diagnosis within 21 days of having any one of these vaccinations, the COVID-19 vaccination or these others like influenza or the others that you mentioned. Is that correct?
That is correct. Yep. And, with the Trinetics database, you're able to look at the common procedural technology codes. And that's a way we can really look at these vaccination procedures in a much more detailed fashion than you might be able to do in other sort of areas like claims database. Because at the time, as you remember, people really weren't claiming vaccinations being performed. They were just being performed en masse, sometimes in free clinics, sometimes in churches and other locations where there wasn't a good collection of data so we could determine really who the populations were that were receiving this vaccination.
So you're pretty confident in the accuracy, let's say, of who had this particular vaccination and when they had it. Is that right?
Yes, absolutely. There's no question about the data with regards to its ability. There are drawbacks to any study you do. I mean, this is not the entirety of the U.S. population. We looked at patients who received vaccinations, and vaccinations did change over time. As you're aware, there are 2 or 3 other different types of non-mRNA vaccines for COVID-19. We weren't able to compare those. We were able to look at the mRNA vaccine in this case, just because of the way we selected our patients for this study.
So what did you find out? How did the rates of retinal vein occlusions compare across the different vaccinations that people had been receiving?
So we did look at using propensity score matching. The relative risk for new RVO diagnosis after the first diagnosis of COVID-19 was not significantly different than after influenza and after the tetanus, diphtheria, and pertussis vaccinations. We did see, however, that there was a greater relative risk compared with the second dose of COVID-19 vaccines, about a 2.25 higher rate and a range of about 1.33 to 3 was the relative risk rate of a higher rate of potentially having that RVO after the second COVID-19 vaccination.
So not after the first one, but after the second one. Can you explain why that may have been? Was somebody's immune system revved up to then have that happen? Or is it just coincidental? If you have a second vaccine, you're going to pick this up in somebody?
There's a couple different things that I think filter into this that I believe are sometimes biasing this population. The first is that there was a hesitancy to receiving the second dose, so there's a smaller population of patients that we're testing, and so therefore there are uncontrolled differences in the patient populations, potentially differences in recording events potentially between both events that occur. But you're absolutely correct. That concern was there with regards to revving the immune system up, potentially increasing the physiological risk. Initially, you may be naive to that immune response, and then you're more likely to provoke a hyperviscous state following the second immune response and therefore the RVO. But again, looking at the numbers of patients, we're looking at very, very small numbers across the board. The absolute percentages of the total population having this, we're in the 0.003% to anywhere from 0.004%. So very, very small percentage of patients, 104 patients out of the 3 million that we looked at actually had an event. And I treat retinal vein occlusions all the time, Neil, and this could be explained by a lot of other baseline factors. There's probably a lot of other things that we take care of, diabetes, high blood pressure, other conditions which have a higher consideration for relative risk than this. So thinking about this in a weighted format, this really is not a lot of weight to say that there is a huge weight to potentially this as a big driver for RVO, more so than some of the more common things like diabetes and blood pressure that we see and take care of all the time.
So I think you're saying if it does happen, if it is increased, it's got to be pretty small and maybe only related to a second dose. And even if it is increased, we're talking about very tiny numbers, like, fewer than maybe 1 in 100,000. Is that right?
Absolutely. These numbers are very, very small that have the absolute event. And so at the end of the day, I think the readers of this article, and hopefully the vast majority of the U.S. population, should feel that these risks of these mRNA vaccinations, again, was driving some of the fear around this. This is one of the first times we've used mRNA vaccines in this wide population-based approach to helping individuals with this condition. Prior, it had been inactivated protein fragments and things like that were presented to the patients for the immune response. But this is truly different, right? We're giving them mRNA to then transcribe a protein to be at risk to then create the immune response, which when you look at this and the approach for it may be considered a larger humoral response than we would have seen with just inactivated proteins per se. But yet, no matter how you look at this, the numbers are incredibly low and incredibly small.
So how confident are you that when somebody said retinal vein occlusion in this database, it really was one? And is it possible you missed some retinal vein occlusions in people? Maybe they weren't symptomatic, maybe they didn't come in, maybe it was misdiagnosed as an artery occlusion.
Yeah, so we know that there's always a disconnect between ICD-10 codes and what happens in real-world practice, and we know that there's probably some disconnect there. And the other maybe drawback we had with our study was that we did look at 21 days following the initial vaccination event, but we did not look 60, 90, 120 days later. So is it that there was a delayed, potentially, immune response and we missed that? That's certainly a concern, and that's certainly a drawback of this study. But yes, you're right. All of these things potentially may have led to either a slight over or under reporting of this. But in general, we think that ICD-10 codes match fairly well with what we see in our practices in general. I would also say that when you look at some of the confounding patient characteristics, we can't necessarily find those within the database itself. It's extracted EHR data, but it's not perfect. And so things like body mass index, history of smoking or tobacco use, history of prior CVA. The biggest risk for a stroke is a prior stroke. So I think, again, those comparisons we cannot make within this database. But yet, when you look at the numbers in total, the numbers are really quite low and slightly reassuring given what we found on the study.
Also, your event rate is this retinal vein occlusion, which even for our medical students, they often know what that looks like, this blood and thunder appearance. So I have to believe that most of this maybe was accurately diagnosed, but there are other subtle retinal vascular occlusive events that have been suggested to have been associated either with the virus itself or with vaccinations following it, or both. For example, this acute middle maculopathy, the middle layer vessels occluding. Any clues on whether that maybe has the same bias as this perception that some people had that retinal vein occlusions were associated with this? Because that would require perhaps a more subtle diagnosis to look for in these big data sets.
Yeah, absolutely. So I think your point is well taken that certainly those things like acute middle maculopathy have been shown to be related to those. But nonetheless, when they show a lot of those pictures, I think a lot of us miss those diagnoses. Those are very subtle findings. As you stated before, RVO is one thing that we pretty much can pick up with good reliability. AMN is something that I think a lot of retina specialists, even given a blank slate of an OCT that clearly showing some of those initial changes, may miss if they didn't look just outside the macula in those patients. This is a rich database. We can ask a lot of questions. We may miss diagnoses that are there, such as AMN and even subtle RVOs, but yet big signals that are present are going to help us, and this is one where we didn't see a big signal with this at least event.
And you compared this at least to other vaccines and their event rate with retinal vein occlusion. Is it possible all of these vaccines potentially increase the risk a little bit of retinal vein occlusion? And so you didn't see a difference among them, but they all do it a little bit.
And I would say to that still in 3 million doses, the numbers of patients, there were around 100 or so patients within the trial that were in the diphtheria, pertussis vaccination area. There were now probably 100 or so patients in the influenza vaccination area. And think about that for a moment. Influenza is one that is given even widely, if not more widely, and over many years compared to these mRNA vaccines. And we should have seen some sort of increased rate. But if you look at some of the influenza numbers here, it was 93 patients over the entire course of the study that was completed over a few years. So you're right. It's hard to compare absolute numbers. However, the absolute numbers are so small that I really wouldn't write home about this at this given stage. I think this is a nice study just to show that, those initial reports, which I'm glad JAMA Ophthalmology steered away from, could have really been out there and pointed to an event which really just was a coincidental report and did not add to the scientific literature like this study did to really give us a good sense of really there was true event rates that were higher than other confounding factors that might be present.
So what would you advise the patient now that walks in with a retinal vein occlusion and had a vaccine recently and asks you, did the vaccine do this?
So this study was actually spurred on just by one of those patients, actually. I had a young woman who came to my office who presented after a COVID-19 vaccine and was told by her neurologist that the retinal vein occlusion, which was diagnosed by another ophthalmologist, was absolutely related. And I was in the position as the clinician taking care of this young woman in her 30s, telling her about the risk of this and the unfortunate thing where there were case reports present. And so we had to go out and do this study. This is how actually this whole study began in the first place. And so I would say to them at this point in time, while there may be an association with COVID-19 and some of these thromboembolic events, that there does not appear to be an overwhelming risk of concern with the mRNA vaccines. And I would not advise somebody to consider the risk of this as being a concern to them with regards to not having the vaccine, given the certain thromboembolic events that we've heard of and seen of in the first wave of COVID-19, as well as the other waves that occurred subsequent to that. And I think that should be the hopefully take-home message for these patients who might be listening or reading this article.
Well, that's great. I'm Neil Bressler, and I've been speaking with Dr. Rishi Singh, the president of Cleveland Clinic, Florida, about the risk of new retinal vascular occlusion after mRNA COVID-19 vaccination within a very large electronic health record data set. You can find a link to the paper in this episode's description. Rishi, thank you for sharing your information.
Thank you.
And thank you all for listening to this JAMA Ophthalmology podcast. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. This episode was produced by Shelley Steffens at the JAMA Network. The audio team also includes Mary Lynn Ferkaluk, Audrey Foreman, Lisa Harden, Hannah Park, Daniel Morrow, and Dr. Linda Brubaker, Senior Editor for Multimedia. Thank you all for listening.